Hi, dear students. So, welcome to this session where we are starting with Prof Exams Made Easy with the DocTutorials. My name is Dr. Amit Jain. I am MBBS and MD in Biochemistry. And guys, today we will discuss that how to approach the upcoming Prof Exams, right? So, probably all of you are already done with your second term exams, right? And you have got your results in your hands, right? And now you are having a lot of course, right? That you have to finish in the few days, or I can say in just 1 or 2 months, right? And you have to revise and also finish whatever is left. Like probably the molecular biology part is not much covered by you, right? It has been covered in your exam in your university, but not by you, right? So that is what I mean to say here is that about the revision part that you will be able to do very easily. But first, I believe that you should target all the unfinished tasks, right? So the unfinished task right now, you may be having what molecular biology probably, right? And some extra topics like maybe the topics of immunology, the topics of let's say the cancer. Okay, so these are the topics like that you have not covered in the recent exams. So pre-prof is coming probably in your August, right? And then September you will be having your prof exams. Fine. So definitely you're left with less amount of time, but yes, biochemistry is not that vast compared to your anatomy, right? So definitely you can do that. And guys, in the prof exams, right, the most of the examiners, like even I'm the examiner, so I'm also working with some medical college. So what are the topics which are favorite of the examiners, which are very frequently asked in exams, that we should know first in hand, right? So for that, guys, what we need to do is that take the biochemistry papers, right, from your seniors, right? And for the last 4 to 5 years especially, because all this the changes in the course that happened after 2019. So you should have ideally all the papers which started from let's say 19, 20, 21, 22, 3, 4, and 5 you are giving, right? So at least 4 to 5 paper sets you'll be having. So just look at them, and when you look at them, you will find what are the most frequently asked questions in the exams from the different topics. Though I will also tell you all the topics and all the important concepts that you should learn before you reach your examination hall. Fine, guys. Okay, so now, guys, just remember one thing that the course, right, may be definitely vast, but it is easily coverable, right? You can easily cover it. It's not something that you'll not be able to cover. Okay, so all the three subjects are important and never leave any subject, I mean, like less important. Okay, don't think that anatomy is vast, so I'll do that more. Then physiology is little less vast, and biochemistry is again little less vast, right? So all the three subjects are important. All the 19 subjects that you will read in your MBBS, they will all be important, right? During your exams, during your practice in your life. So okay, so guys, so now what I start here is in the prof exams. Let's begin. In the prof exams, guys, what are the questions that you're getting? So in the prof exams, we are getting the questions like one part that is of MCQ based. Okay, so most of the time the MCQs that you're getting, okay, in your exams, they are very simple, right? They're very simple and they're very straightforward. Hardly few questions will be the clinical scenario based. Otherwise, you can have the questions like, let's say, what is the rate limiting step? How much ATPs are used or produced? What is the enzyme deficient in phenylketonuria, or any topic, any question, right? And then you may have some clinical scenario based questions like the patient presented with this and that, and the patient's muscle had a large quantities of glycogen present, and patient had, let's say, the muscle weakness and exercise intolerance. So what is the name of the enzyme deficient? So they have already given you the hint that it's a glycogen storage disease. Okay, and the muscle weakness is one of the common feature, and so probably it may be McArdle's disease, for example, right? So that's how you will get the questions, right? Then you can get the questions in MCQs again, which are based on diagnosis, which are very less frequent, I believe, right? So diagnosis means like suppose they have given you the case, right? And then they will ask you that what is the name of the disease that you are studying in the exams, right? So like in the previous question, it can be, let's say the name of the disease is McArdle's disease. Instead of asking the name of enzyme deficient, they can ask you the name of disease itself, right? So that is again the type of question in the MCQs that you'll be getting. Then because you're reading biochemistry, you may get certain questions based on laboratory values, right? Like patient presented, a young patient came to the hospital with chest pain, and he was found to have a let's say some ECG changes, and his cholesterol level was very very high, maybe let's say 400 mg per dl. Normally it is less than 200 mg per dl, right? So you should also remember certain values, not much, right? Some few values which are taught in your syllabus, right? Like cholesterol, HDL, LDL, triglycerides, urea, creatinine. But even if you don't remember, it doesn't matter, because in your MCQs, you'll be getting the values. The examiner will also give you the values that this was a normal and the patient had 400. So that is like hypercholesterolemia, right? Okay. So you may get the questions which are based on values. So you have to identify the case. Okay. So that is like, MCQ based exam questions that you'll be getting. Okay, guys. Now, the other type of questions that you'll be getting is a long answer question, which sometimes students find tough, right? That, don't just look at the length of the question. Questions are very easy. You know why? Why the questions are very easy? Because they're not just asking one thing, they're asking the multiple parts of the same question. I'll discuss one question today. Like, for example, we'll discuss a topic which is one of the very important topic, like urea cycle. So long answer questions are never like a long answer. Like they don't expect you to know only one thing. They expect you to know multiple things in the same topic. So even if you don't remember some steps or some particular clinical scenario, you will still be able to answer the other points, right? So just don't leave it unattempted. Obviously, you will not leave it unattempted, right? Then we have short answer questions. Guys, short answer questions can be sometimes a straightforward question, right? They can ask you the direct question like, explain the, uh, let's say, the functions of S-adenosyl methionine, right? Or explain the metabolism of methionine with and enumerate the functions of SAM, that is S-adenosyl methionine, right? That's a methyl donor. For example, I'm converting norepinephrine to epinephrine, for converting for making of creatine, for let's say converting serotonin to melatonin, right? Okay, like that. So just remember. So that is like short answer questions, like number one type. Then second short answer type of question can be reasoning type of question, like explain. Okay, explain like reasoning based question. Okay, explain all the whys, like why this, why that, like why the patient of homocystinemia, okay, have increased risk of heart disease, for example, right? Why the patient with cystinuria has kidney stones, okay? Why the patient of phenylketonuria has mental retardation, okay, or having a mousy odor in the urine. Fine. So they may ask you the reasoning based question, which are still short answer. Like they may be the question of just 2.5 marks. Okay, reasoning based. Otherwise, they can be a question of 5 marks. Long answer question can be of around 15 marks with the multiple sections in that I'll discuss also today. Then there can be question of clinical cases, which is based on clinical cases that again I'll discuss today. In the clinical based question, guys, they will ask you the question in such a way. Let me show you, right? That suppose like this, a newborn that developed seizures 2 days after the birth was diagnosed with elevated serum ammonia levels, right? Glutamine levels and alanine levels. Neonatal urea cycle defect was suspected. So already they have given you the diagnosis that it's a case of urea cycle defect, right? And the urine analysis was performed, right? For the orotic acid, and the serum was assayed for citrullinemia. And the levels of both were found not elevated, right? Read this, not elevated. Okay, so they can ask you like a defect in which enzyme? we are talking about, defect in which enzyme can cause such symptoms, why uric acid levels are normal in the above case, and in which case they rise and why they rise, right? They can ask you like that. And name one drug, name. So they're asking you just a name, so stick to that name, right? When they say in the exams, they can ask you the question enumerate, right? Or name, right? Let's say if I ask you enumerate the urea cycle disorders, so you just have to enumerate. And if I ask you enumerate the urea cycle disorders and their deficient enzymes in a tabular form, you just write the name of disease, hyperammonemia type 1, and the name of enzyme deficient CPS1. So guys, it is like that, right? So don't write unnecessary because you have a long paper. You have to finish all the questions, right? So don't get overwhelmed, right? I know this question, I will write more about it. So because then you'll not be able to attempt other questions, though you have a lot of time, but still. Okay. So guys, stick to the point. And suppose if you don't know much about a particular question, right? You know a little bit, but you know the clear-cut point. Huh? Suppose if I ask you, like, what is the activator of urea cycle? Okay. And so you know the N-acetyl glutamate. So just highlight it. Okay, like if I ask you, like, why there is uric acid urea in the type 2 hyperammonemia, so you know the reason. So, and you have less time, so just write the most important line and highlight it. So guys, always highlight. It is very important in the university exams or in your prof exams to highlight the most important things that you want to show to your examiner that yes, I know the crux. I know the most important point of this particular question. Just don't write everything, because no examiner is interested in your stories, right? Okay, there are a lot of students and examiners don't have that much of time to read each and every line that you're writing, and sometimes these lines may have no meaning. So if you want to, get the good marks, always highlight. Like activator of urea cycle is N-acetyl glutamate. Highlight this. If the question asks how it is formed, highlight this. If the question asks like enumerate the urea cycle disorders, so just highlight the topic also. Disorders of urea cycle. Then mention one, two, three names, whatever it is. If they ask three names, write the three names. Okay, and just don't mix everything. Always, once you are having, suppose there are three questions here. So once you are done with your question number one, give little space, right? Give a space of 2 lines, 3 lines. Then start with the question number two. Then start with the question number three, right? And highlight that I am mentioning now question number three. Okay, that is very important, right? So that it doesn't get missed by the examiner. Fine. Okay. So every section of the question you should mention in such a way that it doesn't go unnoticed, right? In the exams, right? So your hard work should be highlighted. What I mean to say, correct? So your hard work should be highlighted, right, guys? So this way you can, attempt the question. Like, now, guys, what are the other types of questions, right? Viva questions. So obviously they're not in the theory exams, but definitely they will be in your practical exams. So guys, Viva, when you are appearing for the Viva, definitely the Viva practicals, and there will be Viva for theory also. And guys, again in the Viva, the questions that the examiner asks you will be very easy, definitely, and they will be based on your knowledge, right? Like reasoning, for example, like enumeration, for example, right? I will not ask you the name the enzyme deficient in von Gerke's disease, for example, right? So, but I can ask you like name the disorders which are like, type 1, type 2, type 3 glycogen storage disorders, right? Or the which glycogen storage disorder doesn't affect the muscles or doesn't affect the liver, fine, like that. Which pathway, if it is not working, can cause hypoglycemia? Which enzyme deficiency, right, in a particular pathway like gluconeogenesis is causing the most severe type of hypoglycemia in the newborn babies, like glucose 6 phosphatase. Okay, so be ready for that. I will not ask you the enumerate the steps of glycolysis, right, in the Viva. So when you're preparing for Viva, just go through the topics and, find the unique point. During the theory revision also, find that unique point. Okay, this can be asked in exams. Like why ammonia is toxic to the brain. This can be asked again in the Viva also. This can be asked in theory also. Why? So always be ready with your whys. Be ready with your, key points. And guys, some of the topics which are favorite of the examiners in the Viva are like vitamins, minerals. So never miss them, right? Vitamins are not just vitamins, they are the whole biochemistry, right? When I talk about thiamine, it's not the berry only, which I mean, I should remember. When I talk about thiamine, I should know that it is the most important cofactor or coenzyme for pDH reaction. This is how your Viva will proceed. Okay, thiamine, what is the most important function? And you will say it is the activator of Krebs cycle and pDH reaction, right? Now the next question will be, let's say, on the pDH reaction. Then the next question may be on the Krebs cycle. Then from the Krebs cycle, I can ask you the question on, let's say, inhibitors, right? So you should know the vitamins and minerals and vitamins, very important for Viva. You should know, guys, something like inhibitors of poisons names. Okay, like cyanide is an inhibitor of complex 4 of etc. Or if I ask you, let's say the ricin, okay, from the castor seeds is an inhibitor of protein synthesis. And how does it work? That I can ask you, right? Some basic drugs, if it is given in your basic books, that you should also know. Basic drugs. I'm not, saying like, statins inhibit HMG co-reductase. That can be part of your Viva, but not much will be asked on the drugs. But inhibitors, vitamins, okay, and diseases, right? Diseases. So they should be, by heart. Okay. And guys, because you're reading biochemistry, the rate limiting steps, okay, rate limiting steps and their regulation, that should also be very, strong hold. You should have a very strong hold on these topics. Rate limiting steps and regulation, diseases, enzyme deficient, and if you know a particular symptom, well and good, right? And if you don't know, doesn't matter, right? I am not expecting you in the first year knowing the clinical picture, right? Most of the things that you should know is the name of the disease and the one at least basic gross clinical picture. If you know, that is fine. Like, in the PKU, for example, phenylketonuria, there is mental retardation. I don't expect you to know like why there is hypopigmentation, for example, like why there is, let's say, the scissors in the patient, etc., etc. But you should know why there is mental retardation in the phenylketonuria. Okay, you understand my point? Like why there is, nervous system failure in a patient of maple syrup urine disease, right? Or scissors are there. Why there is stones in the patient of cystine urea, right? Because cystine is insoluble and it precipitates, right, in the form of hexagonal crystals in the kidney. So like that. Got my point, guys? So these will be the type of the questions in the Prof exam. So again, be prepared for your MCQs. Long answer questions are mostly easy because they are made of short answer questions. They're not actually long answer questions, right? They are multiple short answer questions clubbed together. Most of the time you'll see that. I'll show you now. So let's see, like for example, there is a question. Okay, so okay. Now guys, first of all, most frequently tested topics, because we will be taking the 2 sessions on the amino acids, right? And I will cover a lot of important things in the amino acids that you should know in the Prof exams. So most frequent topics in the amino acid section in the long answer will be urea cycle, right? Then the disorders of methionine, phenylalanine, tyrosine, their complete metabolism with the steps, right? You should know the steps. Like, tyrosine metabolism, if we start from phenylalanine, it makes tyrosine. Tyrosine then converts into, let's say, para-hydroxyphenyl pyruvate by the help of transaminase enzyme, etc., right? Finally, I'll be getting fumarate plus acetoacetate in the last step. Which enzyme is absent in L-captonuria? Which enzyme is absent in tyrosinemia? Right? So this will cover everything, right? What are the products of tyrosine? So like if I'm studying tyrosine, I should know products of tyrosine, 2 disorders of tyrosine, the steps of tyrosine metabolism, right? And then I'm done, right? So tyrosine, phenylalanine, and methionine like are one of the most important topics which are asked in exams. Okay, methionine metabolism is also linked with two other disorders like homocystinemia and cystinuria. So the whole metabolism includes a lot of diseases. It includes SAM also, acidonosyl methionine. So you should proceed like this, that if I'm studying methionine, I should know these points, right? I should know the basic steps. I should know homocystinemia. I should know cystinuria, their basic things, their steps, their management, their enzyme deficient, etc., etc. Any particular vitamin involved, okay? Any particular coenzyme involved, like in the metabolism of phenylalanine, BH4 is involved, for example, right? That is tetrahydrobiopterin. Okay. Then guys, we should know special products of amino acids and their importance. Like histidine makes histamine, and what is the importance of histamine? Like that. Okay. So glutamic acid makes GABA, arginine makes nitric oxide. So we should know their importance also apart from the reaction. So special products, they can be part of, the long answer questions. Then guys, we should know all the inborn errors of metabolism, right? Amino acid ureas, and special focus on, I told you all the disorders, right? They're favorite of examiners, especially homocystinemia, PKU, phenylketonuria, L-captonuria. These are one of the most important questions in exams that you'll be getting, right? Okay. So these are the most important long answer questions that you should know. And there's in the short answer questions. See, short answer questions can be the part of long answer question. I told you already, long answer questions are always in the chunks. They're not like, if I give you a long answer question, I will not ask you only urea cycle, explain. What explain, right? So that will be just part of question. I'll show you. So explain the steps of urea cycle. That is one question. Then I'll have multiple other questions in the 15-mark question. Okay. So vitamins involved, SAM, I told you, transamination, disorders, I told you, and why ammonia is like, why questions, I told you, short answer questions. So these are one of the common questions in the amino acid topic that can be asked in exams, right guys? Okay, so let's take this question, guys. Fine, let's take this question. Can you see this question? So suppose this was a long answer question you get in your exams, right? So actually this is the same question I gave in the exams, right? In the last year, Prof Exams. And it was like exactly same question. So explain the steps of Urea cycle and its regulation. So here we have 6-mark question. Steps and regulation make 6 marks. So you have to draw the Urea cycle and then write about regulation. So that makes total of 6 marks, right? And you can imagine like this, that 5 marks for the drawing of urea cycle and explaining some points, like how much ATPs are used, this was a rate limiting step, etc., etc., right? Then explain why ammonia is toxic to human brain. Okay, that is second part, 3 marks. What is the link between urea and Krebs cycle? 2 marks, right? So that means this question is not very important. I mean, like you have to just write the name of the links, or if you can explain and if you have time, that is fine. Explain any 2 disorders of urea cycle, 4 marks, right? So guys, in this case, what you can do is, if you don't know the explanation, explanation, this is like a question which will give you more amount of marks, okay? Because it is 4 marks question and you have to just mention the name of disorders, right? And the name of enzyme deficient. And if you know some clinical point, that is fine. Otherwise, it's even okay, right? You will get the good marks here. So this, don't leave this, right? Okay, this is the more scoring portion. 4 marks and you have to just mention 2. So you can mention 4 also. Yeah, you may get little better marks, right? Fine. So let's take this question, guys, now. So today we will discuss one topic that is called as urea cycle, right? That is very, very important topic. So let's talk about urea cycle. Fine. So guys, first thing, what is urea? Urea is the excretory product, right, of the nitrogen. See, the point is that human beings, okay, human beings, they cannot store nitrogen. They cannot store the nitrogen. So nitrogen means amino acids, isn't it? So we cannot store amino acids. Can you store? No, you cannot store. You cannot say like, if I'm sitting and if I keep on consuming lots of amino acids or proteins, I'll be able to, make muscles. No, no. I cannot make muscles when I'm sitting all the time. So I need to do exercise to do more muscle or protein synthesis. Daily protein synthesis, that is okay, that I need to do, right? For that I need amino acids. But what if I'm eating in excess? I cannot store them. See, if I excess carbs, if I consume, I will store them in the form of glycogen. As you all know, we can store them in the form of glycogen. So guys, we can make glycogen from the carbohydrates, right? Or we can even make fats from the carbohydrates, right? And fats can also be directly stored as triglycerides, but we have no mechanism to store the amino acids directly. So guys, that is why this nitrogen, which is a part of amino acid, as we all know, amino acid, right, is made of two things, amino group and acidic group, right? So I can use all the carbons, right? Carbons can be utilized by human beings as a source of energy, but I cannot use this nitrogen. So guys, this nitrogen has to be excreted out, right? So in the form of urea, we are excreting it. Human beings, we are excreting in the form of urea. That is 2 moles of nitrogens, NH2CONH2. Okay. So that is a urea molecule. So now you understood that why we need to make around 30 grams of, why we are excreting around 30 grams of urea per day in the urine, right? Why our liver is making this all the time? Because nitrogen is toxic to our body in the form of ammonia, right? Ammonia we cannot handle, especially our brain cells cannot handle the ammonia. Right, guys? So that is why we have to remove it. So some organisms can remove uric acid, right? And some organisms can remove ammonia, like fish, for example. So, but humans cannot remove the nitrogen directly. They need to convert first into urea. So, guys, to convert the amino acid into the urea, we need to follow 3 steps: transamination, oxidative deamination, and finally, right? So first step we always do is called as transamination. Okay, transamination can be a short question also, guys, right? What is transamination? Transamination is a transfer of nitrogen from one amino acid to the other keto acid, right? For example, if I take alanine, right, which is amino acid, and if I take alpha ketoglutarate, which has no nitrogen, so it is something which is a what? Which is a keto acid. So it has no nitrogen. Okay, so guys, we can use alpha ketoglutarate in most of the transamination reaction as an acceptor of the nitrogen, right? And there's the enzymes that we use are called as transaminases. Okay, transaminases, they are dependent on pyridoxal phosphate. While reading vitamins, you should know the active forms. Like what is the active form of pyridoxine? Pyridoxal phosphate. Like that. What is the active form of B5? Pantothenic acid. Answer is coenzyme A. You should know the active forms. Right? Some vitamins may not have the direct active forms, but yes, a lot of them have active forms. Right? Like what is the active form of vitamin K? Hydroquinone form. That is active form. Epoxide form is inactive form. So we should know the active forms. Hope you understood, guys. Okay. So transaminases are B6 dependent. For example, I will call this ALT. And what I'll be getting, because I have removed nitrogen from alanine, I got pyruvate. So guys, this pyruvate is all the carbons, no nitrogen. No? So from the alanine, I made pyruvate. And now I can use this pyruvate as a source of energy. So I will not waste the carbons, only the nitrogen I cannot store. So that we have to remove. And what I get is, guys, glutamate. Okay, glutamic acid or glutamate, right? So guys, there are 2 terms, glutamate and glutamin. So this is glutamate, first thing to remember. Now guys, we can take any amino acid, like if I take oxaloacetate, for example, okay, if I take oxaloacetate, then there's this enzyme will become AST, and this will be, sorry, this will be oxaloacetate, and if I take aspartate, okay, aspartic acid, then I'll be getting oxaloacetate, and name of enzyme is AST. So this is how we can convert every amino acid to glutamate. Now see what is happening. Any amino acid coming, alpha kg is taking nitrogen from them, right? So alpha kg is, storing nitrogen from everywhere, taking nitrogen from every amino acid and converting into glutamate. So all the excess amount of nitrogen in the human body is now appearing in the form of glutamate, isn't it, guys? So glutamate is formed. Fine. So what is the next step, guys? Next step. So this was a transamination, and if we talk about the next step, this is called as the oxidative deamination, right? So guys, when you start with the urea cycle, you should mention at least like this. Definitely steps are most important, but you should start like this, that nitrogen is toxic. Why? Because it is ammonia. Then how to take the nitrogen from every amino acid to glutamate, and glutamate then release the ammonia in the liver only. So this oxidative deamination, let's say, occurs in the liver cells, mitochondria, and guys, we will be taking this glutamate dehydrogenase enzyme, glutamate dehydrogenase, and I'll be converting this back to alpha ketoglutarate plus the ammonia, right? So this ammonia is released. This ammonia is toxic, and glutamate dehydrogenase. Guys, glutamate dehydrogenase is the enzyme, right, which is unique because it can use both NAD, right? So right now, let's say it is using NAD and converting this to NADH. Fine. But in the reverse reaction, guys, this can use NADPH. Okay, NADPH. So NADPH. So it can use both. What is the unique point about this enzyme? That it can use NAD also and can use NADPH in the reverse direction also, right? Okay. So when I want to assimilate ammonia to glutamate, I can use glutamate dehydrogenase again, right? So that is called as oxidative deamination. Guys, whenever we use the term oxidative, that simply means that you know there is involvement of some electrons, right? Oxidation. That means electrons have been removed. Or here we can say the dehydrogenation happened. Dehydrogenation is also a type of oxidation, right? So most of the time, all the dehydrogenases are causing oxidation, but here it is deamination also. Like pyruvate dehydrogenase was oxidative decarboxylation of pyruvate. It was like that. Correct, guys? So this is oxidative deamination. So this ammonia is released. Now this ammonia is very toxic. So guys, then what we will be doing? We will be converting this into urea, right? So in the urea cycle, we know the steps that, right? I need not to explain you, but carbon dioxide binds with ammonia. First enzyme is carbamyl phosphate synthetase 1, and this is the rate limiting enzyme, right? Rate limiting enzyme. Carbamyl phosphate is formed. This binds with ornithine, and name of enzyme is ornithine transcarbamylase, second enzyme. And this forms the citrulline, right? Citrulline now appears in cytoplasm. So guys, this pathway takes place in mitochondria in the pink, and now this is cytoplasm, right? In the blue, as it is shown here also, color coding. So citrulline then binds with aspartate and converts into argininosuccinate. That's an enzyme called as argininosuccinate synthetase. Then argininosuccinate lyase or argininosuccinase, both the same names, makes the fumarate plus arginine. Fumarate will go to the TCA or the Krebs cycle. So guys, this is one of the most important link of urea cycle and Krebs cycle. That is a part of our question also, right? So this will go to the TCA cycle. And that you have to highlight, you can highlight here also during exams that, okay, this goes to the Krebs cycle. And you can write here in the bracket link of urea and Krebs cycle, right? So you have already answered, but you can again mention later on. But that means, guys, that is highlighting it 2 times and obviously makes the, getting good marks, right? I mean, like getting good marks simply means what? That you know the concept. Point is that, right? You're not writing stories. You are writing the concept. You're writing the stuff that is most important. So arginine finally acted upon by the last enzyme called as arginase, and this makes ornithine. So and urea also. So as ornithine is recycled, and ornithine is something which is kind of in the urea cycle, we can call it as a catalyst, right? Basically, it is not used and it is not giving its carbons and nitrogens and making the urea. It keeps on recycling, right? And it will go back to the mitochondria by the ornithine translocase. When it is going back to the mitochondria, it is going through a transporter called as translocase, right? You know what is translocase? Translocase is nowadays called as a class 7 of enzymes, right? That is like, coming this side and this side. So going both the direction. That's called as translocase. Clear, guys? So that is the urea cycle, very easy, 5 steps. Right? You can say total 5 steps only. And then we have to mention everything about it. Okay, so what we can remember, what is the rate limiting step? So guys, answer is CPS1, right? What is the obligate? So guys, you should know this term right now. Obligate allosteric activator. Okay, obligate allosteric activator. Guys, what is the meaning of allosteric? You all know that something which is binding to the enzyme at a different site than the active site and can activate or inactivate the enzymes, right? Suppose this is enzyme. Okay, this is enzyme, right? And this is my active site. This area is active site, and this area is called as the allosteric site, right? So active site and allosteric site. There are the two sites we have. So allosteric site is a site. This is a site. This is allosteric site where the N-acetyl glutamate will bind on the CPS1, and this will activate it. So guys, obligate means 100% required. Guys, in the biochemistry, only two names that you should know for your first year exams. That what are the two obligate allosteric activators? This can be a Viva question also. Two obligate allosteric activators for urea for in the biochemistry. One is N-acetyl glutamate for CPS1 of urea cycle, and second is the acetyl CoA for the pyruvate carboxylase in gluconeogenesis. Pyruvate carboxylase in gluconeogenesis is activated by acetyl CoA. That is an obligate allosteric activator. Understood what is the meaning of obligate? Obligate simply means 100% required. You know that, right? Simple English word. Obligate. Okay. So that is your rate limiting step and obligate allosteric activator. So you understood, guys. So you should write that. What is the name of obligate allosteric activator? N-acetyl glutamate. Guys, it is formed with the help of acetyl CoA and glutamic acid. And this is in the short, we can call it as NAG. So if I take acetyl CoA plus if I take glutamate, and the name of enzyme is NAG synthase. NAG synthase, as you can see here, right? NAG synthase. So this will be making me what? This will be making me the N-acetyl glutamate. And this, so this is the most important activator. So even the deficiency of this enzyme can cause the diseases like urea cycle disorders, right? Okay. So this is the second thing, guys. How many ATPs are used? So we have not covered yet, but CPS1 is synthetase. Now, synthetase means the enzyme which is using ATPs, right? So synthetase, they will use 2 ATPs. CPS1 use 2 ATPs. And there is argininosuccinic synthetase, which was adding the aspartate. That is also using 2 ATPs. So total of 4 ATPs are used. Some book says the fumarate will goes to the Krebs cycle and it is, producing malate, and this has also generated one NADH. Because fumarate was formed now, fumarate goes to the Krebs cycle, makes the malate. Name of enzyme is like, malate, then malate goes to the oxaloacetate, and this makes NADH, one NADH, and NADH gives me 2.5 ATPs. So this will be minus, I can do minus here because fumarate is giving 2.5 ATPs. But guys, that is a long distance relationship, right? Okay, we are not going into that deep. You should write that how many ATPs are exactly used in urea cycle. Answer is 4. But you should also know this concept if your examiner asks you, if they ask you in the Viva, for example. So 2.5 minus may happen if I go that long distance, right? Okay, so 2 ATP plus 2 ATP, 4 ATP total used, right? What is the rate limiting enzyme? CPS1. What is the regulator? I told you. What are the source? source of nitrogen in urea. So guys, what are the sources of nitrogen in the urea molecule? So first nitrogen was given by ammonia itself, right? It was ammonia itself. And second nitrogen was given by aspartate, right? We added aspartate now. Don't say arginine, right? Arginine was made in the cycle. It is not added from outside. So arginine is not used here directly. So we don't say arginine. Arginine itself was made by aspartate. Okay. So aspartate is the second source of nitrogen in the urea molecule. And there's what else we should know. The other question was link, right? Link of urea cycle with the Krebs cycle. So the first answer is, best answer is fumarate. Okay. But have you seen the question? It was mentioned links. So there should be multiple answer, na? Links. So fumarate is the answer, right? It will go to the second is guys aspartate, right? So if somebody asks you what is the name of the amino acid linking urea and Krebs cycle, answer is aspartate. Why? See, because guys in the Krebs cycle, suppose when I have oxaloacetate, this oxaloacetate can be used using AST and can generate aspartate, and this can go to the urea cycle. Or I can reverse it also from the urea cycle. Urea cycle had aspartate, right? We are using it now. So, but basically this is a link. Krebs cycle, oxaloacetate, aspartate, transaminase makes aspartate, goes to the urea cycle. So that is also the link of urea cycle and the Krebs cycle. So guys, basically the Krebs cycle's derivative is also used in making urea molecule, right? That's why it's a link, right? So that is a link. Fine. So guys, then there was a question in our, if you look at the question again, let's see. If you look at the question again, it was like this. So, just a second, this. Okay. So the question is, explain why ammonia is toxic to the human brain. So what is your answer? So guys, first stick to the biochemistry. I have seen some students, they are writing XYZ anything, right? That they have read, in some books or online or anywhere else, right? And they will open the chat GPT, right? And during their studies, not in exams. And then, they will write directly about NMDA receptors in the brain are, excited by the glutamate, causing excitotoxicity. Such words, right? You don't have to write them. First, stick to the basic biochemistry. Okay. So why, like the question is, why ammonia? Okay. So why ammonia is toxic to the human brain? Fine. So the answer is, the one answer that you should highlight, okay, is that first of all, ammonia easily cross the blood-brain barrier. So guys, ammonia will easily cross the blood-brain barrier, right? And in the brain, so when it reaches the brain, okay, so when it is reaching the brain, it will bind the alpha ketoglutarate. So ammonia bind with very high affinity with alpha ketoglutarate of, where is alpha ketoglutarate? Of the brain's Krebs cycle. Fine, so it's like this, reaction is like this. Ammonia plus alpha ketoglutarate of the brain's Krebs cycle makes glutamate. Okay, it makes a glutamate. Now guys, this glutamate can also bind one more ammonia and can make the glutamine, right? So guys, this we are doing every day. You know why? I'm not talking about toxicity. Let's forget the toxicity right now and just understand that this is happening in our body every day because ammonia is reaching the brain every day. How? Because it may escape the liver metabolism, or there's a lot of ammonia is absorbed directly in the blood, right? From the, let's say, from the intestine, because the gut microbes, they are making ammonia all the time, right? So that is like the ammonia which is reaching our liver every day, which is something different, which is not derived from directly amino acid metabolism. This is the extra source of ammonia we are getting from our gut. So guys, these gut bacteria, which are very important for our health, because they are, maintaining our immunity, they are making some substances which are decreasing inflammation in the intestine, even in the brain. So they are definitely our helpers. Okay, but they are also producing ammonia, by the various putrefaction reactions and their own metabolism. So this ammonia may reach brain, and this we are doing every day. To detoxify it, I have to convert this into glutamine, right? And this glutamine will go to the liver and make the ammonia and then urea that I'll explain, right? Okay. So let's say this glutamine is formed, but the first thing to remember is, so the excess ammonia deplete the brain's Krebs cycle alpha ketoglutarate. So the excess amount of ammonia depletes the brain's Krebs cycle alpha ketoglutarate. Clear? And guys, because of this, there is decrease in Krebs cycle. If I say there is decrease in Krebs cycle, and that means there is decrease in ATP production in the brain. And guys, this is causing neurological clinical features that may be like scissors, convergence, etc. etc. Okay, so guys, that may cause a death of neurons, right? The failure of the brain, coma ultimately, right? So guys, you understood this is the point that you should mention in your exams first and highlight it. Rest all for you right now are secondary. Definitely, guys, because of increase in glutamate, just I'm mentioning, but this may not be even in your basic books right now. But glutamate, you know, bind, okay, binds the brain at certain receptors known as NMDA receptors, right? And this causes the excitation of the brain will be increased, and this causes a neurological death of neurons, basically by various mechanisms. So I'm not going into that details, but brain bind NMDA receptors and cause the neuro or excess glutamine, no excess neurological damage, right? Neurological damage. So guys, hope you understood this point. So that's all about the why ammonia is toxic to human brain. So this is the definitely the one of the theory which is nowadays most accepted, right? Okay, apart from this basic knowledge, definitely this is the basic knowledge, and this is definitely the most important point also because it is depleting ATPs. But this is the second theory, and nowadays, the most accepted theory, right? Apart from the previous one. Okay, that you will read again in your maybe pharmacology later on or medicine later on, right? So that is all about why ammonia is toxic to brain. Now, guys, why it is not toxic? Because we make glutamine, right? So, guys, ultimately what happens is this glutamine. So, if we forget the toxicity, this glutamine will goes to the liver, and this liver has one enzyme called as glutaminase, right? So, this will release ammonia from the glutamine, and guys, from this ammonia, I'll be making urea. So, this we do every day. So, this glutaminase enzyme in the liver is again very important, right? So, guys, if I ask you the question that okay, what is the carrier of ammonia? Yeah, if I ask you the question, the non-toxic form of ammonia transport, right? Non-toxic form of ammonia transport, and your answer is glutamine. Guys, glutamine is so common in our blood, right? Because we keep on converting the excess on this like unnecessary ammonia to glutamine, and this glutamine will go back to the liver to make ammonia or to make, I mean to say, for urea finally, right? Actually, one of the very common amino acid in our blood is glutamine, right? Because it is a carrier of ammonia. All right, guys. So non-toxic form of ammonia transport, storehouse of ammonia is glutamine, right? Okay, so these are the points that you should understand and mention in the exams. Okay, so what are the questions again? what is the rate limiting step, how the urea cycle is regulated, what are the steps of urea cycle, why ammonia is toxic to the human brain, right? Okay, and what is a non-toxic form of ammonia transport in a healthy individual? Answer is glutamine, right? Okay, glutamine. So guys, these are the points. And link of urea and Krebs cycle I have already explained, right? Okay. And there's now explain any two disorders of urea cycle. So that you should remember that urea cycle has multiple disorders, right? What are the disorders of urea cycle? So guys, just one by one. So CPS1 deficiency, you can say, or the N-acetyl glutamate deficiency. This causes the type 1 hyperammonemia, right? Then second is ornithine transcarbamylase second enzyme deficiency. This causes type 2 hyperammonemia. Guys, in type 2 hyperammonemia, there is increase in ammonia plus there is increase in orotic acid, which may appear in the blood or urine. So guys, what is this orotic acid actually is? That, imagine the second step is missing. So the first step was going on. So what is the first step's product? Carbamyl phosphate. As this carbamyl phosphate will enter into the pyrimidine synthetic pathway. What is the rate limiting step of pyrimidine synthesis? CPS2. So rate limiting step of the pyrimidine synthesis is CPS2. But if ready-made carbamyl phosphate from urea cycle, which is now deficient, is going to the pyrimidine synthesis will start, and this will make orotic acid. And there's this orotic acid will convert into uracil, may convert into uracil, not always. So I know you are already done with pyrimidine metabolism, correct? So in the pyrimidine metabolism, now orotic acid was one of the steps. So how it is forming from the carbamyl phosphate now? So carbamyl phosphate of urea cycle will appear because of this enzyme deficiency, and it will appear in the pyrimidine synthesis, and this will make excessive orotic acid or maybe uracil, right? But uracil formation is less. You know why? Because to make uracil or UNP, you need pentose sugars, which are not easily available all the time. They are made by a junction, which is not active. Only because of this enzyme deficiency, carbamyl phosphate enters into pyrimidine synthesis. So guys, that is our second point. So guys, you can easily diagnose if only ammonia is high, it is type 1. Ammonia plus orotic acid is high, it is type 2. Then citrullinemia, that is the next one. So you know the name of enzyme, argininosuccinate synthetase. Then argininosuccinate aciduria, that is the fourth one. Argininosuccinate lyase is the name of enzyme. Then number 5 is hyperargininemia, right? Hyperargininemia. And guys, this is due to the deficiency of arginase. Okay, arginase. Right. Fine. Guys, as you go away from the first step, you will have less symptoms in your patient. Why? Because ammonia toxicity is not decreasing now. Because we have converted, let's say in the hyperargininemia, arginine is high. So we have already converted the excess amount of ammonia into arginine, which is about the soluble amino acid. So when you go away from the first step, obviously the toxicity of ammonia is decreasing because we are making some soluble products like citrulline, like argininosuccinate, etc., etc. Right? And there's a last but not the least is triple H syndrome, where the one H is hyperammonemia, second H is hyperornithinemia, and third is homocitrullinemia. And this is due to the deficiency of ornithine translocase, which is sending ornithine back to the mitochondria in the last step. So if ornithine is not going back, this ornithine may appear in blood, hyperornithinemia. Correct, guys? And hyperammonemia, obviously, urea cycle disrupted, and homocitrullinemia, because carbamyl phosphate in the first step. Imagine if ornithine is not reaching, right? Let's say here, imagine that ornithine is not reaching. Okay, so this was your ornithine, right? Let's say This is the ornithine. Na? So if this ornithine is not going back, and carbamyl phosphate will accumulate. So carbamyl phosphate will bind with certain amino acids like lysine, and this will make the homocitrulline, right? That's a different term, homocitrulline. So hope you understood, guys, right? Okay. So that is your triple X syndrome. Guys, how will you treat urea cycle disorders? Obviously, give low protein diet. Low protein diet you have to give. But guys, you can increase the levels of arginine, right? Arginine activates the N-acetyl glutamate synthase. That means, there is increase in the CPS1 activity. So arginine is beneficial in most of the cases, but don't give in hyperarginemia, right? Already arginine is high. So arginine can be given. So low protein, why? Because protein only makes the ammonia, amino acids here. So what else you can give, guys? You can give certain drugs. So I have shown you that clinical question. So guys, there is one drug that's called as benzoic acid, right? Benzoic acid. So this is a drug that we can use in urea cycle disorders. Why? Because this benzoic acid binds with glycine, right? Guys, glycine has a nitrogen, no? And this will make a non-toxic hippuric acid. So hippuric acid is non-toxic, and this will appear in the urine. So guys, basically, what is the funda? What is the concept? If the patient has high ammonia, that is nitrogen, take the nitrogen out in the urine anyhow. If we are unable to make urea, make something else and take the nitrogen out. So guys, we took glycine out, nitrogen, glycine out. Glycine is a non-essential amino acid. So even if we decrease it from the blood, our body will make it again. So obviously what we did is we took the nitrogen out, right? So ammonia production will decrease because we are now the rest of the nitrogen will be used in making glycine. So we have taken the glycine out. Correct, guys? So this is benzoic acid that we can use as a drug. We can also use phenyl acetate, which binds with glutamine, right? And take it out in the urine. So we can use one more drug, that is phenyl acetate. And this will bind with the glutamine. So this makes phenyl acetyl glutamine. And this is non-toxic, and this will appear in the urine. Now, guys, glutamine has two nitrogen, so two nitrogen will appear in the urine. So this is how guys, the scientists, doctors identify that what can be the other channel of nitrogen removal, and that's how these drugs are being utilized or created. So we understood. So we understood this point, guys. So this is all about guys, urea cycle, and hope you understood that how you have to answer the various questions on urea cycle, and especially the long answer question, right? Long answer question. And guys, in this clinical case, if you see, what is the answer? Now you can answer this clinical case now. So in this clinical case, a defect in which enzyme? So guys, which enzyme defect? So a defect in the defect in which enzyme? So only ammonia is high, na? So what is your answer, guys? Tell me in the, if you are listening, then can you answer in the comment section? The answer is CPS1, right? CPS1. Okay, because only ammonia is high. Now, rest of the question try. Why uric acid levels are normal in the low case? Because OTC is not deficient. Uric acid levels rise only in the OTC deficiency. So I told you why, right? Because carbamyl phosphate, which was accessed in urea cycle, enters into the pyrimidine synthesis, and uric acid is one of the intermediate of pyrimidine synthesis. Correct, guys? Name a drug which is used in management of urea cycle. Answer is benzoic acid. You can answer, or the phenyl acetate also you can answer. Two answers you can write. Correct, guys? So see you. I'll see you in the next session, right? We will discuss some disorders of amino acid metabolism and the questions related with them. Fine, guys. So good night and start preparing. Thank you so much. Bye bye. Okay.